Good morning. Thank you for your presence. I would like to thank Alejandro and Ernesto and my partners and colleagues from CRF for organizing this event. At CRF, we've made great effort and we've been making great efforts for education initiatives, moreover in Latin America. We believe that there is a great value, and moreover in Argentina and in many Latin American countries as well, that we made use together and we made tap greater. And this small and modest educational course, as Ernesto has said, is very important. And in fact, it implies engaging our centers in Argentina into international studies in sort of in a two-way road, from there to here, from here to there. In the next 15 minutes, I will be discussing with you how at the CRF we looked into the process from the preclinical stage, as Juan has mentioned, from first in humans to first. We are going to make a difference between two different types of clinical studies. First, where we have a randomization. I'm sorry for my Spanglish. When there is randomization of patients. And the others, the first are called clinical trials, and the others are sorts of investigation studies where there is no intervention and they are just clinical studies. Among these studies are the ones that have been mentioned by Juan, the so-called preclinical studies and with animal models. The FDA and the NIH sometimes discuss and argue and keep defining the different studies, but we follow the definition of the NIH that divides them into 5 categories or groups: the treatment trials, prevention trials, diagnostic trials, screening trials, and those that are focused on quality of life. Among these 5 groups, we have the phase 1 to phase 4 studies, and then clinical trials. I wanted to highlight this because oftentimes there's confusion. We talk about a clinical trial or a study, or what kind of study we can undertake, whether it is just a registry or whether it is study where there will be intervention involved. And oftentimes the 5 kind of studies are mixed up with the different phases. So we will be starting from clean slate so as to start working on this educational course. Sometimes we also confuse, and myself confuse. Therefore, we have the phase 1 to phase 4 studies, and we will stop for a minute there. I will not be briefing you about each one of them because all of us may read this on a web page or on the literature. But what I will be discussing with you today are the challenges that we face as clinical investigators for each one of the phases in the studies. And Juan has talked very thoroughly about the first in human and the challenges posed to the clinical investigator, the main investigator or primary investigator. There is where the risk will be assessed. You need to have open eyes as to where the device is, which status of development is, whether it is in the preclinical stage, whether they are bringing a protocol to our center. It might involve very few patients. Our center might be asking us for 1 or 2 patients, and we may find it quite easy as investigators because, who can't submit 1 or 2 patients? But these are high-risk studies. And then, as Juan has mentioned, we need to look at the technical dossier, all the explanations, discuss with the sponsor. As investigators, raise our questions and our doubts. The phase 2 study, whether A or B, those are larger studies or trials where, apart from safety, there is an associated risk. They have between 200 and 300 patients. One of the endpoints is efficacy. Therefore, here not only do we have to consider for the enrollment of patients, those that have high or low risk, but also we will be considering results as to efficacy. Phase 3 and phase 4 studies or trials are larger. Many of them are randomized. And the challenge we face as investigators in our center is, will I be able to contribute with a number of patients I am being requested, and does it match the characteristics of my center? Will I be able to devote as much time as necessary? Because this is another challenge that we face as investigators, and this is related to raising the standard of the trials we are undertaking. The investigator is not only the one signing the contract with the sponsor, but he is intimately or closely related to the quality of the results. Therefore, in this stage we have the challenge whether if I have 300 patients, will I be able to devote as much time as necessary, and so on and so forth. And those are trials where safety is one of the endpoints, but of course efficacy is very important, and there are other endpoints to be taken into account. There might not be the primary endpoint, but secondary features or characteristics that should be taken into account in connection with the device or drug. The success of this process, moving from the preclinical stage to the marketing stage, lies on the protocol design. We always highlight and emphasize, and CRF, moreover, specializes in designing scientifically detailed protocols, but we can't forget that protocols have an associated methodology that needs to be considered. And when we have as a challenge designing this protocol, we need to bear in mind all these methodological points to be taken into account. And the most important thing is to understand what phase I'll be in, whether phase 1, 2, or 3, the way in which the device has been developed or the drug has been developed, and our aim. Historically, what has been easier to us, and not only to us, but to other investigation centers, are the classical protocols where we have either phase 1, phase 2, or phase 3. But a very interesting question was raised. I mean, is there any way in which it can skip one of these phases and move from a phase 1 study directly to a pre-marketing approval stage? And Juan has answered very well, but I want to add something else, that the answer is no, you can't skip any stage. But what we are working on very hard is try to design protocols, that they are the so-called adaptive protocols, that don't let us skip phases or don't let us bypass anything, but throughout the course of the study. And this is a very important difference with classical studies. We may adapt or adjust the protocol as we learn. I mean, from what is happening, we may move one patient from one arm to the other, or change treatment, or modify the dose. And this was impossible before. We needed to wait for 5 or 6 years till the study finished in order to have the results and the outcomes and the publication to be able to bring a patient in or not. And throughout the course of the trials, we realized that we learn more from the mistakes that we have made, and we are more capable of correcting them. And I have listed some of the issues permitted by adaptive designs, like having more flexible hypotheses, non-inferiority or superiority hypothesis, and be able to change them, redefining the inclusion or exclusion criteria, combining different phases and treatments, and also being able to stop the study earlier if necessary. And of course, the beauty of all this is to have a plan B, and it does not fail right in the middle. I mean, as we have seen after 5 years for a protocol, and they are left there. In the next 5 minutes, I will be talking about what it is for me, not only because I lead this area. I will be talking about adverse event reporting. This is one of the major challenges for investigation protocols, and you cannot hide the elephant within the bathroom when we have adverse events. And I want to mention this because not long ago, in 1986, less than 1% of the problems were reported by investigators. Nowadays, it seems unacceptable, completely unacceptable, that very short ago there were people that, though they realized that there were adverse effects and our patients were suffering from these adverse events throughout the course of the trials, did not report them. And among the cardiovascular studies, we'll be assessing, of course, the classification of the adverse event, the seriousness, the causality, whether it is likely related or probably related, and possible and probable. I did not include both classifications because that is related to the time of occurrence, from the time the procedure was initiated till the event adverse occurred. Are they not related? I mean, is this expected or not? And I've highlighted all these features because on the basis of this classification, we will be issuing the report to the FDA. And these report levels and the characterization of adverse events is something that Liliana was asking here, and I find it very important, and I will be briefly sharing it with you, what has allowed us to have better quality studies and safer studies and be able to understand things better and to redefine pathologies. In fact, we are sitting at the panels and boards that keep defining and redefining the meanings of these adverse effects, thanks to data collection. Therefore, who do we have to report to? To the ethical boards and the regulatory agencies of each country. If I am participating in an international study, I need to talk to the sponsor. The sponsor is responsible for this, and I have to resort to him, and he needs to report to international agencies. Moreover, the sponsor is accountable for reporting those adverse effects to the clinical event committees in charge of assessing these adverse effects. There is a very detailed list and a very thoroughly studied list when we need to report, after how many days. I need to sort of I can shelve this, and then I can say, well, let's report over the weekend. No, I can't do that. That is absolutely prohibited, and this has regulatory implications and success implications. As I've said before, the thoroughly detailed protocol includes adverse effects reporting. And what shall I report? Well, that needs to be detailed in the protocol, and that is very important as investigator to ask and to talk, to discuss to the sponsor or whoever it is, whether it is a sponsor of a pharmaceutical company or a governmental agency. Of course, serious adverse effects. Who should I report to, and what should I report? And the endpoints of the study, who should I report them to? I mean, the regulatory agency and my ethical committee, or to somebody else's? All these details are related to the success of the clinical research. Of course, the endpoints are always reported, and that should be reported to the IRB, to the sponsor, and the trial assessment committee. In the studies that we work with here, and this is to the interest of the CRF and CASI, we know what to be reported, when to be reported. Either they are primary endpoints or secondary endpoints. And to conclude, what I want to tell you is that all the management of all these adverse event reports leads to the success of the clinical trial based on its quality or the completeness of that adverse effect report and the integrity of the reports that I have submitted. And this is no minor issue. We've had many failures, and we've faced big failures in many famous trials that Argentina has participated in, and it has been very difficult to draw information or data from studies that have costed more than $80 million because the report quality was very poor, because they were incomplete, and because the document integrity was not credible. They had no date, patient ID. Therefore, if you invest money and then as investigators, and I wear both hats because I am two sides of the street. Oftentimes I participate as sponsor and as an investigator, and sometimes I wonder, why can't they complete this? And then when I participate as an investigator, sometimes I wonder, why didn't they like what I sent? Because I've sent everything. And you need to be very meticulous, to be very detail-oriented, and we need to demand our coordinators to be as obsessive as they can, so as to submit in a good fashion the adverse events. And well, everything has evolved. I mean, we are moving from paper-based reports to electronic-based reports. Therefore, the process would be more agile, and this would help us to reinforce the concept of adaptive protocol. If I have more adverse effects in real time, I can redesign my study in real time as well. And of course, the impact has been fundamental, because in the last few years, thanks to a good data collection, we've been able to redefine effects such as myocardial infarction with enzymes, with different enzymes, how to prioritize one or the other, because we've been reporting many data. At the beginning, it seemed ridiculous. Why do you want troponin at 0 time, 5 hours, 10 hours, 24 hours? Thanks to that, we've been able to sit down and redesign the guidelines for the definition and being more accurate and bearing a greater impact. Thank you.
¿Qué tal? Buenos días.
Good morning. I would like to ask you, when you were speaking about data collection, that this was migrating to an electronic format to be able to collect them. Do you use an ad hoc interface, something for registry collection, or understanding that you use HRs as EHRs? And for example, if the data, if this is quality or safety points, do you get it from the clinical record, or is it necessary to enter an interface? We work with both. We can just enter the electronic system of the center with which we are interacting, and we have people who are specialized in these sites, a group that we also have an electronic system, and we ask them to upload the information, so we prepare the dossier and we include all the information. But there is not a difference as far as quality. Either work very well. Well, we are always pressed by time. As principal investigators, we are responsible to tell this to the coordinators, but sometimes communication fails. Alejandra, many times the protocols are perfect, and investigators, for example, EVA-3S study is carotid angioplasty. They had to suspend it because of the poor results, and they had proctors with them, and these were the patients that they contributed with. In this sense, should we do an analysis of the investigators so as not to have these sort of mistakes as in EVA-3S? Thank you for the question. It's very important. And when we start a study, we send a qualification form of the site, and in this form we have the population to be studied, the type of population that the site believes is going to be possible, and we have this type of patients. Yes, no, it is very comprehensive. And what are the possibilities that you have of doing this and that study? So I believe that the initiation of the site, when we go and submit the protocol and discuss it, is very important. What we see is an evolution as far as the possibility of saying, well, before clinical trials contributed with huge opportunity. Now, taking into account strict regulations, we are more sincere as investigator as far as what we can do. Good morning. What do you think regarding regulatory times in Argentina? I believe that ethics committees vary. I say it very humbly because I don't know all of them. And I would divide this between the regulatory agency and ethics committee of the hospital. And this varies a lot. We have centers in Argentina that are very quickly approved, and the regulatory agency is the ANMAT. It's more difficult. It's slower. But the idea is to, well, as it happened with the FDA, if an operator from the FDA falls out of the window, is going to fall down a month later, and this was modified really. With ANMAT, it might happen the same. Alejandra, I would like to ask you regarding your idea and your current point of view as far as education in Latin America, and that is how you introduce yourself. What do you consider Argentina from a regional standpoint? That is regarding data quality, volume of patients, experience of prior participation. We had a very strong growth mid this decade, and then this growth was slowed down, also because other regional countries improved their quality of work and the regulatory laws. They have legislation of clinical investigation. We are still developing it, and there are decentralized standards as health care is. But what do you think about it? How do you see us, Argentina, as a country to investigate? I would like to invite Liliana to answer this question, who has much more experience than I do. I humbly want to say that I think that there was a cycle of romance with clinical trials in Argentina, and then based on some experiences we had with some studies that were difficult to interpret, the result from Argentina, and the intensity was slowed down. But in India, China, Japan, Brazil, and Mexico, it didn't happen the same. So to do international trials, even though 75% of data generated in clinical trials is outside the United States, so the emphasis that we are placing is not only from CRF, but also all the companies are looking for international sites. So I believe that we are recovering this. This is one of the possibilities that we have to become better. I have a great hope that we are able to deepen phase 1 seminar, phase 2 seminar. Today we spoke about an overview in 15 minutes, but can we organize a talk or a webinar in our site? Education is a priority, and I think that Argentina is on this pathway, definitely. Thank you, Alejandra. I think that the romance ended mid last decade, but this was basically because that it took longer than 9 months for Argentina to accept any clinical trial, unless we had a way to be able to go forward. I would not like to speak deeply about it, but I would like to say that in Argentina, investigators really became frustrated. There was very good education, also very good participation. The good clinical practices were done very well, but today there are no courses on good clinical practice in Argentina. I don't know who in Argentina is involved in investigation. I participated in the last course 2 years ago. I am a coordinator, etc. But what I would like to say is that Argentina really had a negative impact as far as quality, because clinical trials were not approved, and everybody became exhausted trying to attempt it. So it went down, 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 and quality was worsened. But however, as we coordinate a clinical trial, we were able to include a very large number of patients. We were in the international average, or even better as far as the quantity of restudies, and as far as quality, we were over 90%. So in the last 10 years, in the last decade, this was the average, as you might remember. So I think that this is because of the lack of authorization. And Anmand, with Dr. Chiale today, is trying to look for a better result. We had this imprint that in the Anmand that is published in the newspapers that, for example, clinical trials killed people. And, for example, we are using vaccines to prevent hepatitis. However, newspapers published that people were killed. There was an investigator that did not have the approval of the fathers to use the vaccine. Well, they skipped a step, 5 or 6 cases. However, the mass media took this, and the vaccine was blamed. But this is the vaccine we're using today in Argentina and in the world. What I would like to say is that there was a poor advertising in investigation. And I remember with the freedom trial with the ANMAT, and the Argentina also participated a lot. After Brazil, it was the country that had the largest number of subjects. And I remember that in the middle of freedom, all patients had signed the informed consent, and we had the red seal, which was an awful seal that patient to whom you offered surgery and angioplasty, which is a standard method, there was a red seal stamped in the informed consent, where you had to call the patient back and show them the red seal that was practically telling them that they were kind of a guinea pig. So this red seal was a problem, and it lasted 4 years, and it really made clinical trials be very, very difficult to be made in Argentina. The idea is that this is trying to be reverted. What I would like to say is that we have to attend courses, and in order for Argentina to be prepared to be able to do high quality clinical trials. And I think they are going to be back. Yes, I do hope that. And the final comment is thank you for your comments. Of course, they are very valuable, but I believe that this is an experience that it has been lived in the States, not in the last decade, but before, and some of the things we learned is that when, well, we really put a lot of pressure in the FDA when we submit a protocol. We invite the first line of investigators, we discuss, and also the soundness of knowing each step of the protocol, and really we put a lot of pressure on the FDA. That is why it is very important to understand our role as principal investigators, which is really very important. And education starts from the principal investigators